Hello everyone, my name is Dabholkar, my roll number is 6204, and my topic for assignment is process of prokaryotic translation. Initiation is the first step of translation. And there are three initiation factors involved in this process. First one is the IF1. Second one is the IF2, which is a GTP-bound factor. And third one is the IF3. The ribosomal RNA rRNA present in cytoplasm is made up of two subunits, the 50s RNA subunit shown in blue color and the 30s RNA subunit shown in pink color. There are three major sites on rRNA that are a site or acceptor site, P site or peptidyltransferase site, E site or exit site. IF3 is bound to E site and it does not allow large 50S and small 30S subunit to bind together. IF1 is bound to a site and it inhibits binding of tRNA and large 50 subunit. IF2 comes and binds to IF1, which is GTP-bound factor. IF2 brings charged formylmethionine along with tRNA bound to it. The tRNA binds to the P site of rRNA. This structure formed is called as 30s initiation complex. Due to binding of initiator tRNA, there will be conformational change in 30s RNA amino acids, which will result in release of IF3 and IF1. Once the IF3 and IF1 are released, the large 50S subunit along with mRNA binds to 30S subunit. When this happens, GTP on IF2 is hydrolyzed to GDP and it is released. The complex now formed is called 70S initiation complex. Next is elongation, and the factors involved in it are EF2 or elongation factor thermostable, EFTS or elongation factor thermostable, EFG or elongation factor translocase. All of these elongation factors are GTP bound. GTP-bound elongation factor EF2 brings the charged amino acid tRNA at the A site of ribosome. Once the tRNA binds, the GTP is hydrolyzed to GDP and EF2 is released from ribosome, and there is a peptide bond formation between amino acids which are on a site and P site. GTP-bound EFG helps in the translocation, As it binds to the ribosome, the GTP is hydrolyzed to GDP, and as a result of this conformational change, the tRNA on a site carrying two amino acids shifts to the P site. The elongation of translation continues in this manner until the stop codons are reached. The EFTS helps EF2 in the recycling of GDP by GTP. Next step is the termination, and the release factors involved in it are RF1, RF2, which are class 1 factor, and RF3, which is class 2 GTP bound factor. Once the stop codons reaches the A site, the translation machinery now proceeds for termination, in which first the RF1 occupies the A site. The three amino acids GGQ present in RF1 helps in release of polypeptide chain from ribosome. This release is triggered only when GTP-bound RF3 gets associated with RF1. GTP is hydrolyzed to GDP, which finally releases RF3. Once the polypeptide chain and RF3 gets dissociated, RF2 binds at the A site. In presence of EFG, the RF2 releases the two uncharged tRNA from P and E site. When finally IF3 binds to the E site, the translational machinery gets dissociated.